Good afternoon, everyone. Um, my name is Rudra Sawant, and I'll be presenting the paper Hierarchical Clustering of Corals using transfer learning. This is my master's thesis that I've worked on for the last 1 year. I was helped by my supervisor, Gianluca, and Tom Bridge, who is the expert on corals from the Queensland Museum, who also supplied us with the coral dataset that we'll be using in this paper. Okay, so what are corals? Corals are basically a type of species which are mostly found below the sea level in the oceans and sometimes in the creeks. They come in different forms. They have different structures. It's very difficult to identify which corals belong to which class, or which two corals are similar or which two corals are dissimilar. So this is one of the major problem in the classification of coral taxonomy. So for many years, researchers have been trying different methods to classify corals and make a hierarchy of their taxonomy, meaning basically to find out which corals are similar to which other corals and which of them belong to which subclass, which genetic phylum, or which clade or subclade. Okay. So for many years, what researchers have used is they have used human experts to manually look at these coral samples and classify them according to what they see. So basically, they used to use the morphological similarities, which are basically the skeletons of the corals and how the structures of the skeletons are to classify them. But recently, people have used genetic detection techniques where they actually compare their DNAs or genetic material to actually find out which corals are similar or dissimilar. And these two techniques are having many inaccuracies and inconsistencies between them. And since this has been done, we need to find better ways to classify corals and have a better taxonomy of coral species. So one of the ways that people have been trying is by using image classification techniques, because recently image classification using neural networks has gained popularity in and can actually classify many of the images as good as humans can do. So that technique is what we'll be using in our paper. So our aim is to basically classify corals into a taxonomy, basically hierarchical classification of a coral, using the Queensland Museum's coral dataset. So they have provided us a dataset which we'll be using for this, and we are using image classification techniques, which are neural networks. And then what we are doing basically is first training a classification neural network, image classification neural network, in the task of classifying corals belonging to different categories. And then once the network is trained, we use this network to obtain feature embeddings of each coral images. Basically, these feature embeddings are used as a representation of each image. And then we use hierarchical clustering algorithm, that is hierarchical agglomerative clustering, to cluster these feature embeddings into a hierarchical taxonomy. So previously, people have done similar works on a dataset called the MLC 2008, which is a similar coral dataset, but we didn't have access to this one. But they've used pre-trained models like VG16 and DenseNet to classify corals, and they've achieved pretty good accuracy. Other work was done on the same dataset that I'm using by Jaden and Gianluca and Tom. They have actually used Lime explainability layer to assist experts into classifying corals, where the experts could actually see which parts of the corals was used by the model to classify them into a particular category. Okay, so the dataset that we'll be using is supplied by the Queensland Museum. It has 8,745 coral images, which belong to 106 different coral categories. So some of the images are displayed on the side. So these are different categories, and we have 1,600, 1,006 such categories having images. Some of them have many images, like 15, 16 images, but some have very few images, like 1 or 2 at max. And these images are of different kinds. Some of them are of specimens which are taken in a lab environment where there's like dark background and only the specimen is visible, and that photos are taken. And some of them are actually taken in the ocean environment where you can actually see the sea floor and the blue water, the blueness of the water and all. And you can actually see the coral as whole instead of the actual skeleton structure inside. And some of them were also taken under a microscope where you can actually see the intricate details of the structure. So these were the 3 types of images that we had, but they were also like skewed. So most of them were lab environment images, like more than 60% were lab environment images, and a few were microscope, very few, and a lot were in situ as well. So we had to pre-process this data. To pre-process this data set, the first thing we did was converted all the images into a standard format of JPG because there were different files, including TIFF and PNGs and other file formats. So we converted to a standard format. Then we resized them to 224 by 224. Sorry, just having an error there. And then we removed the microscopic and in situ images. This was done because due to the different types of images that were there, we wanted the models to be trained on only one kind of images, because the microscopic images and the lab images were totally different. Like you couldn't actually tell the difference, or we couldn't actually compare these 2 images. That's why we said we need to have a standard images that the model trains on, and that's why we used only the lab images, which didn't have any background noise, which didn't have any other objects in it. Similarly, we removed any images with like other objects present. Like some images had rulers, some images have like paper or plastic bags on them. So which made the corals not visible, or we couldn't see the coral properly. Such images were removed. And we also removed the categories of corals which had very few images in them. Like some of the categories had only 1 image in them. So it would be very biased for the model to actually use that 1 image and then train itself to classify all images based on that 1 sample that it has seen. So we removed all categories having less than 3 images in it. And then we use the data augmentation techniques to increase the data set, because our data set is pretty less, because we have 1,600 different classes and 1,006 different classes and only just 8,000 images. So we used data augmentation techniques like horizontal, vertical flips, rotation, brightness, increase, decrease, zooming, shearing, and so on. So after the data pre-processing, the first step was to train the models. For this, we used 3 main models. These are VG16, Inception, V3, and DenseNet. These 3 models were used directly by importing their weights, which were trained on the ImageNet dataset. So ImageNet dataset is another huge dataset which has different objects and different classes of those objects. And the models have been trained on this. So the weights were directly imported and kept as constant in our models. So, and what we did was we followed 2 approaches to train these models. The first approach, we just trained the top layer of these models. So what we did was we kept all the layers, the weights in all the layers as constant, and we just updated the weights in the last layer of these models using the Coral dataset. In the second approach, we just trained the top 2 layers instead of just 1 for the similar task. Okay, and then the results of the first approach were as follows. We got a test accuracy, max test accuracy was around 28.8%, which was pretty bad. But then when we trained two of the last layers, we got around 39.7% test accuracy. So this test accuracy was obtained for DenseNet model, which we thought was the best test accuracy which we got in both the approaches. So we decided that this model performed the best on the image classification task on corals. So we decided to use this model for extracting the features. So what we did was the DenseNet model from the second approach, which gave us the best test accuracy, was used to extract features from each of the coral images that is there in the dataset. So each of these feature embeddings was a vector basically representing that coral image. And we use these vectors for the further processing. So before we use the vectors, what we did was these vectors had huge dimensions, like 51,000 dimensions something. So we reduced the dimensions using PCA. While reducing, we made sure that we preserve 95% variance in the data. And once we had the dimensionality reduction done on the vectors, we use these vectors to classify the corals in a hierarchical fashion. For this, we use Agglomerative Hierarchical Clustering method to cluster these corals. For this method, there are again different criterions that you can use, like this average, single link, complete link, WORDS criterions. These are basically criterions where you can specify how you want to compute the closeness, dissimilarity, or similarity between two vectors. So we use all these 4 different criterions, and we also did one more thing. For each coral category, what we did was we calculated a mean feature embedding, which was a representation of the whole category in one simple feature embedding vector. Instead of having each coral image having its own feature embedding vector, we took a mean of them and then said that this was the mean feature embedding vector for the whole category, and then we clustered them similarly into a hierarchical fashion, so that we have a hierarchy of different coral categories. And the performance of these different clusterings that we did was evaluated using the Sillard score. So Sillard score basically gives you an idea of how much the two clusters, or the clusters in the clustering overlap on each other. So it is between 0 to 1, the Sillard score. A zero means basically that all the clusters are overlapping, and 1 means that none of the clusters have any overlap between them. So for the clustering done on each of the coral image, we found very less Sillard score, but when we had a mean feature embedding taken for each coral category, and then when we performed hierarchical clustering, we saw that average link criterion gave us the best Sillard score of 0.3861, followed by complete link and single link, which were almost close by. So therefore, we decided that the average link hierarchical clustering using the mean feature embeddings would be the best clustering that we could find in this coral data set using this method. And we use that as the hierarchy of the coral categories that we have. And then when we plotted a dendrogram of the coral categories and their mean feature embeddings, this is what we got, how the clustering look like. So these are basically, we don't exactly know the actual subclades or classes that these categories belong to, but what we could find is how close they are or how close they might be. So if you can see, this is the existing biological hierarchy that Coral World website has, Corals of the World website has. So you can see that one of the classes is like Anthozoa, and it has different subclasses, and the subclasses have like clades and subclades in them. So all these are not very consistent with the other this thing, but we could see that there was kind of a structure which was not exactly as what we got, but we could use this to classify corals. So in conclusion, what we got was our test accuracies for the classification task was very less, but around 39% something. And the Sillard score was kind of average, because they say that around 3 to 6 is the average Sillard score for average kind of clustering. So the clustering is not very much overlapping, but it is kind of overlapping a little bit. The quality of, so we could say that the quality of the hierarchical clustering that we have got was kind of average, but still, since we have using this, what we could do is basically we could actually give experts a way to actually find which corals are similar to a given coral image. So this could actually help experts to, instead of looking at a lot of images, they could actually look at a few images and they could actually classify them easily. So basically it makes their job easier. So in future work, we felt that one of the major reasons for getting a bad test accuracy was because there were very few samples for a coral category. Some of them had like 3-4 samples. And even though we use data augmentation, it's a very low number to get to classify such a diverse animal like coral. Also, most of the images that were taken of a coral, they were taken of a single coral instead of different separate corals of the same category. So basically, we need more corals to take images from. Also, what we want to do in future is basically, it would be good to have a model which could accommodate to different coral images taken under different environment, like in situ under the ocean images, which directly could be used to classify that coral into a category instead of just taking lab specimen images. So that's for the future work. And thanks for listening. Any questions would be nice.
Thank you, Rudra, for the great presentation.